Hello, everyone, and welcome to the Melanoma Research Foundation's CureOM webinar on Chemtrac. We are thrilled to have you join us today for this informational webinar about the first FDA-approved medication to treat metastatic uveal melanoma. My name is Sarah Seelig. I'm the director of the MRF's CureOM initiative. My husband, Greg Strax, and I founded the CureOM initiative after he was diagnosed with OM in 2006. When Greg's disease spread to his liver a year later in 2007, and we were told there were no treatments shown to extend life, we were devastated, a feeling to which I know many of you can relate. And now, to start the year 2022 off with the first FDA-approved medication shown to extend life in metastatic uveal melanoma, this is truly an historic moment, and there's so much hope in the OM community. I want to quote a recent blog post by Dr. Keith Flaherty, which you can read in its entirety on the MRF's website. But to quote Dr. Flaherty, in the past 10 years, the treatment of skin melanoma that has spread or metastasized has been revolutionized. Year after year, the percentage of patients with metastatic skin melanoma who were surviving for 5 years and longer climbed by 10% increments. It was an exhilarating time to be in melanoma research. Throughout this decade, in which metastatic skin melanoma jumped from being one of the least treatable cancers to being one of the most successfully treated, uveal melanoma outcomes didn't budge. All of that is changing. We now have a drug, sorry for my emotion. We now have a drug that leads the immune system directly to uveal melanoma cells, regardless of where they may have traveled. In a large trial, this drug, Chemtrac, or Tebi, reduced the risk of dying of uveal melanoma by half compared to any other therapy offered to patients in the other arm of the trial. Now our task is clear. Use the same approach that allowed us to rapidly advance approaches in skin melanoma. This requires continued research and partnership between patients, melanoma researchers, and companies who have treatments that might build on this monumental first step. 2022 in uveal melanoma feels like 2011 in cutaneous melanoma. 2 drugs were FDA approved in that same year, portending the beginning of a revolution that played out over the subsequent years. Thank Dr. Flaherty for his amazing dedication to this field over the years and for sharing his perspective with us. I'm so excited that you are all here today to learn more about this moment in the OM field and about Chemtrac. Education is critical for patients making informed decisions about their care. We are pleased to bring you today's webinar to educate and empower the ocular melanoma community. This session is not only offered live now, but will be available after the live event as part of our video library that you can share. And before I introduce today's speaker, I want to just provide a brief medical disclaimer. We encourage you to use the comment section to ask questions throughout the session. Those questions will be answered towards the end of the session. The information presented during today's session is for educational purposes, and any individual treatment questions should be directed to your healthcare provider. We would encourage everyone to visit melanoma.org to learn more about the resources we have available for our ocular melanoma patients and caregivers. And now it is my great pleasure to introduce our speaker for today, Dr. Marlene Orloff. Dr. Marlene Orloff is an assistant professor of medicine and a medical oncologist at Thomas Jefferson University Hospital, the Sidney Kimmel Cancer Center in Philadelphia. Her clinical and research focus is on primary and advanced melanomas. Her main interest is in rare melanomas, including uveal, conjunctival, and mucosal melanoma. Dr. Orloff's research is centered on investigating the epidemiology of uveal melanoma and the discovery of novel therapies. Dr. Orloff works very closely with the team at Wills Eye Hospital to manage hundreds of patients with primary and metastatic ocular melanoma. Among a variety of many other important roles, Dr. Orloff is principal and co-investigator on a number of clinical trials and is a founding member of CureOM's Vision Registry Steering Committee. We're grateful to have you here with us today, Dr. Orloff, and I will hand it over to you.
Thank you, Sarah. It is absolutely so very exciting today to be able to share information about Chemtrac. As you heard from Sarah, this being the first drug that's been FDA approved specifically for uveal melanoma is absolutely amazing. So I will now share my screen. How do we look, Sarah? Can you hear me?
Great.
Good.
Looks great.
Okay, so everybody, today we will be talking about Chemtrac and everything you wanted to know and more. So a bit just of an overview of the presentation today. We will talk first about how we got here, a little bit about the mechanism of action, some clinical trial highlights, review of side effects, what is HLA and why does it matter, questions to ask your provider, and a bit about what else is out there. So you did hear from Sarah as part of Dr. Flaherty's excerpt from his blog, that the last now 10, 15 years in melanoma has really been revolutionary. The discovery of the immune checkpoint inhibitors has really changed the way in which we treat cutaneous melanoma. And I think when these drugs were first discovered, there was great hope that they would also be effective in uveal melanoma. However, you can see here just a little summary of some of the trials or studies that looked at the immune checkpoints such as ipilimumab or the PD-1 inhibitors, nivolumab or pembrolizumab in uveal melanoma. And unfortunately, you can see here that response rates have never really gotten to where they've been in cutaneous melanoma. Down at the bottom there in yellow, you see the results of the combination ipi-nivo trial in cutaneous, where we saw an overall response rate of 57.6% and a median overall survival of greater than 60 months. So certainly these drugs in cutaneous melanoma have amazing activity. Right above that, you see the largest prospective study in uveal melanoma here with just a response rate of 18% and median OS of 19 months. So, I think over the last 10 years, we've really been trying to ask ourselves, why doesn't uveal melanoma respond the same way as cutaneous? And for a long time, we knew that they were different. But really, I think this discrepancy in how uveal melanoma responds to immunotherapy has really highlighted some of these immunologic differences. We know that there's something called a low tumor mutational burden, meaning the number of mutations we see in uveal melanoma is much lower than in cutaneous, probably related to the fact that cutaneous melanoma is often caused by UV damage. We see something called lack of PD-L1 expression, which is kind of a marker that the immune system has at least acknowledged the cancer. And we all know that when uveal melanoma becomes metastatic, the most common organ it goes to is the liver. And the liver, even in cutaneous melanoma, is often a hard organ to target as it's considered an immune privileged site. So when we think about immunotherapy resistance, there's a couple of different ways you can go about addressing it. You can try to make the tumor more of a target to the immune system. You could try to force the immune system to go after a target on the tumor. Or in some instances, you may want to try to remove some barriers between the immune system and the tumor. And this is where Chemtrac has really identified that second resistance mechanism in its mechanism of action. So here's just a bit of a kind of a busy cartoon on how Chemtrac or tebentafusp works. And so what I like to say, or the way that I like to describe the mechanism of action of this drug is really it acts like a two-sided magnet that on one end binds an immune cell and really drags it over to the cancer. So in this top diagram, you can see what's called a polyclonal T cell, which is an immune cell, and then this big other gray thing is the cancer cell. And basically, the drug is kind of this darker blue little series of ovals here, and basically it acts as a magnet to again bind the immune cell, the T cell on one end, and really drag it over to the cancer cell and force them to engage, and then hopefully the immune system then kills the cancer. The theory is that, while we have a lot of T cells around, they kind of would otherwise just walk right by uveal melanoma. Again, because uveal melanoma may not be as recognizable to the immune system, these T cells really aren't that attracted to it. But basically, again, the drug really drags the T cell over to the cancer. I do just want to highlight just a little bit, because we'll go into this further, that the way that this magnet though interacts with the cancer cell is through something called HLA. And HLA is important in this scenario because the target that the immune system is going after is something called GP100 that isn't necessarily just hanging out on the surface of the tumor. Sometimes it's on the inside and actually needs to be presented to the immune system. And it does through something called HLA, which we'll talk about in a little bit. So now I want to get into some of the clinical trial highlights. So for those of you that's been kind of following along with the story of Chemtrac, it started as a drug called IMC-GP100 way back originally in 2013. It moved into a kind of first in human study in 2015 that actually enrolled both non-uveal cutaneous melanoma patients and uveal melanoma patients. And it was actually in this study that they started to see some responses in uveal. Actually, 2 of the responders were patients with uveal melanoma. And there was this initial thought, which I think no one was necessarily expecting at the time, that, maybe this drug, has some activity in uveal melanoma. And then that is what expanded the phase 1 study specifically in uveal melanoma. So this was what we call the IMC-GP100 102 study. And this was actually 2 cohorts. And we were doing a dose escalation, trying to find the right dose. And then once we found the right dose, we then moved on to a phase 2 expansion for patients who were previously treated. We did see some activity there, and then that study moved on to we call our pivotal study, which was a randomized study, multi-center, international, where patients were randomized in a 2-to-1 fashion to either get tebentafusp at that time versus what's called investigator's choice, where the physician was able to choose either dacarbazine, ipilimumab, or pembrolizumab. And these were patients who were previously untreated. And this was a study that just completed last year that has since resulted in the FDA approval. So I just want to highlight here that the first metastatic uveal melanoma patient actually treated on the 102 study was here at Jefferson in March of 2016. And here you can see some of the team surrounding her. So this was the original study design of the escalation. I just wanted to put this up here because I think a lot of patients may be asking, how did we get to this dose? It's actually kind of a funny dose when you think about it, 68 micrograms. And we actually started at 54, went to 64, then to 60, or then to 73. There were some side effects seen at 73, which is then why we went down to 68. So then the final studies require then weekly infusions. The first week you get an infusion of 20 micrograms, then 30 micrograms, and then 68 micrograms. And then 68 micrograms weekly thereafter. So this is what we call a waterfall plot. And basically it's just showing patients who've had a reduction in tumor size. So on the left here, when bars are going up, that's tumors that are getting larger. And then when you see bars going down, that's when tumors are getting smaller. And then we consider kind of negative 30% to be an actual response. Everything else in here tends to sometimes be still called stable disease, even though the tumors are getting smaller. So this was the original waterfall plot for really those first 19 patients. And I think for a lot of us, this was the first time that we saw a waterfall plot in uveal melanoma, where more of the bars were going down than going up. Dr. Sato actually presented this back in 2018. And this is really what started our, excitement about this drug as a potential option that's going to be effective in uveal melanoma. Not only that, but when we actually looked at overall survival, again, in that early cohort, the median OS at the time of, this data cutoff anyway, which is at 24 months, the median overall survival hadn't even been reached. And at 1 year, 74% of patients were still living. And again, these were patients who, many of which were heavily pretreated, had multiple prior therapies. So, this 1 year overall, survival rate had really kind of blown anything previously out of the water. And then, so again, it was really with that data and then some of the data that we saw in the expansion that led to this, what we call our pivotal study. So again, this was a randomized study, 2-to-1. So you had kind of, so for every 2 patients randomized to Tebi, 1 patient got randomized to investigator choice to kind of increase the number of patients who could get exposure to Tebi. And again, the way that the drug is administered is you get 20 micrograms on day 1, 30 micrograms on day 8, 68 micrograms on day 15, and then weekly after that. And again, this is the investigator's choice. Patients had to be that A201 positive, no prior therapy, including systemic therapy or liver-directed therapy. And what we were really looking for was overall survival. And then actually this unique endpoint of overall survival specifically with patients with rash. And we're going to talk about that in a second. So the results of this study resulted in a publication that many of you may have seen, which looked at the overall survival benefit of tebentafusp in metastatic uveal melanoma. So I just want to highlight kind of what these groups looked like. You know, we have median age here. Most patients were pretty well. A lot of or a good portion of patients had what's called elevated LDH, noting kind of more advanced disease. Here you can see the staging. And then here just to highlight, you know, that a good portion of patients had liver metastasis. Again, as I mentioned, often can be kind of a hard organ to target with immunotherapy. Here's what we call the overall survival curves. So, you know, just the big take home here is anytime that you see curves separate, you know, you can kind of see them separate here. That's a good sign. And here you see the tebentafusp arm underneath the control arm, and you can see that they are quite separated, suggesting that there's an overall survival benefit for tebentafusp over control. And so, you know, when Sarah mentioned that, you know, that Dr. Flaherty had said kind of that half, you know, the risk of death was half in the tebentafusp arm versus the control arm. That has to do with something called the hazard ratio. And then you also see here the median overall survival for the tebentafusp arm was 21 months versus 16 months. So, you know, it was really with hitting this overall survival endpoint that told us that this was a positive study. So the interesting thing is that, you know, for anyone who's been following this drug along is, yes, it's showing survival, but when we look at something called response rate, which is the percent of patients that actually have shrinkage of tumor, it's not a whole heck of a lot. So here we only really see what's called an overall response rate of about 9%, but that doesn't seem to matter. There's a good portion of patients that have stable disease, or we call disease control, so either stable disease or response greater than 12 weeks. But so, like I said, it doesn't seem to matter. You know, the tumors don't need to necessarily shrink, at least by traditional sense, by just looking at scans, for patients to be living longer. And we're still trying to tease out maybe what that discrepancy is. Is there something happening at the site of the tumor that maybe there's parts of the tumor that are actually dead, even though we're not seeing it specifically shrink on the scan? And this was more made evident when we actually looked at overall survival in patients who their best response by scans was progressive disease. So RECIST, reminder, is the way that we read scans for patients who are on clinical trial. We pick specifically target lesions, and then we follow those very specific lesions kind of through the whole disease treatment course. And we're really looking to see the shrinkage or growth of those to determine response. And here you can see that patients, even with progressive disease on Tebi, did better than the control arm that had stable disease. So these, you can see these two lines here. So there was still an overall survival benefit in patients who had progression. And again, we're still trying to tease out, you know, what all of that means. A good portion of the patients on study were actually what's called treated beyond progression. So even if the scan showed that the tumors grew a little bit, patients were allowed to stay on study. This is a phenomenon that sometimes we see with immunotherapy. Sometimes the tumors may swell before they get smaller, or there's something else even happening at the site of the tumor that we haven't totally figured out yet. So now just a bit about side effects. So like I mentioned, this drug is given weekly. The first 3 doses are given in the hospital. So dose again on day 1 at 20, dose on day 8 at 30 micrograms, and then dose on day 15 at 68 micrograms. And the reason that it's given in the hospital is because these drugs, when they basically pull the immune system over to the cancer, not only are they triggering an immune response against cancer, but they can also trigger a general immune response. The other thing to note here is that while the drug pulls the immune system over to GP100, which is something we see on the cancer, GP100 also exists in the skin on normal melanocytes. So the immune system also gets drug over to the skin. So what we really see in the side effect profile, really with those first 3 doses, it's something we call cytokine release syndrome, which is really a term that just encompasses fever, potentially low blood pressure, and potentially problems with oxygen. And a grade 1 cytokine release. So in order to qualify as having cytokine release, even a grade 1, that's just fever. So most patients have a fever. You can see here, not all patients end up having that hypotension. And then the other main side effect we see, especially with those first 3 doses, is rash, because again, the immune system being drug over to the skin can cause various types of rash and also itching is what is considered here pruritus. So we do sometimes see some other things that accompany that, because basically it's like all of a sudden you get a really, really bad flu. So you can see some chills, you can see some nausea, you can see some fatigue. Like I said, that lower blood pressure can be part of that more significant cytokine release syndrome. And then some of the other more rare side effects, sometimes dry skin, which can kind of happen after the rash, vomiting, erythema, so just redness, headache. Occasionally we can see elevations in the liver functions. This may be related to the immune system getting pulled over to that dominant organ that may be involved, the liver, causing some inflammation there. Lipase can be elevated, which is something that happens at the pancreas, diarrhea, lymphopenia, which is a drop in the lymphocyte count, which is actually something that we sometimes want to see on that second day, because it tells us that the lymphocytes are being kind of drug, to its target. Hyperbilirubinemia, low phosphorus, and the opposite of low blood pressure. Some patients have had a little bit of a high blood pressure. So again, this chart kind of highlights, though, that if we're going to see kind of those main side effects of that CRS, cytokine release syndrome, and rash, we really see most of the more serious ones on those first 3 doses. So when we grade side effects, 1 or 2 are more mild, grade 3s are more significant. And you can see that really by week 8, most patients are not having much in the way of side effects at all. So here's percent of patients. So lots in week 1, lots in week 2, lots in week 3, much less by week 4. And by week 4, most patients are actually outpatient. They may get a little fever and a little rash that night, but nothing significant. And then really by week 8 and on, we really see what's called an amelioration, or really a decrease in all side effects. And patients who are out many months, really tend to have no side effects at all. Come in, get their infusion, and then go home. Again, some patients may have a little bit of a fever that night, low grade fever, or some patients can get a little itchy, but nothing really of significance. And then I do want to comment just a little bit on that cytokine release. So, the reason that patients are in the hospital, especially for those first 3 doses, is that, fortunately, we don't see a lot of significant cytokine release syndrome, but there are situations where if the immune system gets too revved up, so high fevers and drop in blood pressures, sometimes we need to use things like steroids or another drug called tocilizumab to kind of basically put the lid on the immune system so that patients don't end up too sick. And so really that's the purpose of those first 3 infusions being given in a closely monitored setting. So the way the drug is now approved is that it requires what's called a 16-hour observation. So when we first were doing the clinical trials, patients were really admitted overnight, but now with the approval, at minimum, patients need to be watched for 16 hours after infusion. Again, really keeping a close eye on that potential for cytokine release. Also, just a little bit about the rash. I would tell patients the rash in some ways can be very predictable, in that the rash would often kind of start maybe about 6 to 8 hours after the infusion. It could be everything from just a little bit of an itch to kind of a full body, sunburn. Patients can be a little, miserable that next day with the rash, but then it's often gone within, 48 to 72 hours. So anyone who's listening who may have, experienced rash from other immune therapies like the immune checkpoint inhibitors, where it tends to kind of just persist as long as you're on the drug, this rash tends to resolve itself with like I said, with about 24, 48, 72 hours after infusion. And then really after those 4 weeks, it tends to go away. So it was really with all of those positive results that you just saw from that pivotal study that led the FDA to approve tebentafusp for unresectable or metastatic uveal melanoma on January 25th. So I just want to talk a bit about HLA and why it matters. So as you even going back here to the press release here, you could see that this drug is approved for something called HLA-A201 positive adults. So what is HLA? HLA stands for human leukocyte antigen, sometimes also called MHC. It encodes cell surface molecules that really allow the whatever cell it is to present bits of antigen, so which is typically kind of foreign things, to the immune system, to T cells. So, here I actually put up a schematic of how this happens with COVID. So COVID can actually get into your cell. There's processes that happen that then allow the cell, the infected cell, to present bits of it to the T cell to hopefully kill it. And it presents it through something called HLA class 1. And so, like I said, the antigen can be infectious like a virus, or it can be a tumor. There's 2 different types of HLA, or what they call MHC. So class 1 is what we're talking about here, which is your A, Bs, and Cs. And then there's something called class 2. And the immune system also uses HLA to differentiate between self and non-self, which is why it's necessary actually for matching in organ transplant. So not everyone has the same HLA for some of this reason. So what is HLA's role in tumor presentation? So like I mentioned, that the HLAs actually present tumor antigen to the immune system, to the T cells, to facilitate the immune response against the cancer. So here you actually have a normal cell, and a normal cell actually may, unbeknown to itself, actually present self antigen, to the immune system. And I'll say, no, I don't want to go after you. You're yourself, so this is the prevention of autoimmune disease. But a cancer cell may present a little bit, which is like a little antigen through this thing called MHC or HLA to the T cell. And then the T cell will engage with it, and then it will hopefully kill the cancer cell. So with Chemtrac, in order to design this magnet that was going to interact with the antigen that's presented by the cancer cell, they essentially had to pick a type of HLA for it to interact with, because it needs to interact with a lock and key in order to engage and kill. And so there are many different types of HLA, but in order to develop the ImmTAC that's able to interact with the presented antigen, they had to pick an HLA that basically was the most common. And HLA-A2 is the most common, found in all ethnic groups. And HLA-A201 is found in about 50% of Caucasians. So that's the story of HLA. That's why you need to be this particular HLA type. That's why they chose this particular HLA type. And that's why if you're not this HLA type, unfortunately, Chemtrac as it's developed right now won't work, because this little magnet, while it might bind, a T cell, it's not going to fit like lock and key on at the site of where the little cancer bit is presented by HLA. It just won't engage, and the drug won't work. So I just want to talk briefly about questions to ask your provider. I know I went through a lot of data here, and some of you have heard about this drug now for the last number of years. But I do think it's important, at this point for for patients to be aware of these questions to ask. And so what I may do is actually just jump right to kind of 9. And then we'll kind of come back to the rest of them and do more of kind of a live Q&A. So, I think, while like I mentioned, the approval of Chemtrac has really, changed the way in which we're going to manage this disease. I just got finished explaining that, well, if you're not A201, then this drug doesn't work. So, what if I have, metastatic uveal melanoma, but I'm not A201 positive? And I'll actually lump in maybe even question 8 here. What if my scans show progression? Because I do want to just mention that there's a lot of other drugs that are out there. For patients who are negative, there's liver-directed therapy, there's still standard of care ipilimumab and nivolumab, and clinical trials. For patients who are positive, there's liver-directed therapy and other immunotherapies, clinical trials, and then there's actually a number of other drugs like IMTACs, like tebentafusp, and actually other immunotherapies that are also HLA restricted to A201. I do want to just briefly mention some observation that we are seeing or have seen in patients who are previously treated with tebentafusp. So this is actually just one example of a patient who actually had prior immunotherapy, had progression in their liver, then went on had immunotherapy, went on Tebi, had progression in the liver, and then we actually re-challenged with immunotherapy and actually had a great response. So, there is some thought with those of us who've given a lot of tebentafusp followed by immune checkpoint, that tebentafusp or Chemtrac, may actually be resensitizing the cancer to be sensitive to immune checkpoint inhibitors. So for a number of our patients who progress on Chemtrac, often our next line is ipi-nivo, because we are seeing some good responses there. So here, and the most amazing thing is this list just keeps getting longer and longer. So these are current clinical trials that are open, most of which are actually open specifically for uveal melanoma. And again, a lot of these trials are available for patients who are A201 negative or A201 positive. We have some targeted therapy trials, some other immunotherapies looking at different targets. PRAME is a study that's very similar, the mechanism of action to tebentafusp, except for instead of going after GP100, it's going after PRAME. There's some other targeted therapies. There's some combination strategies with radiation. There's a few TIL studies that are ongoing, and there's actually a few liver-directed therapy trials that are also still ongoing. So, in conclusion, I just want to say that I think our initial disappointment with immune checkpoint inhibitors really did force us to look and acknowledge what we had known all along, which, was that uveal melanoma is just a very different subtype, and we can't just always lump it in with cutaneous melanoma. We use the failure as an opportunity to understand why. And, IMC GP100, aka tebentafusp, Tebi, aka Chemtrac, is the first FDA approved therapy specifically for metastatic uveal melanoma. And the importance of this can really not be stressed too much or enough. The success of this drug has taught us a lot, but there's still a lot of work to do. I just want to highlight here a patient who, though, got dosed first back in September of 2016, and treatment is still ongoing. So this is a picture of her and her family, I think in a cave in Hawaii. So she's been getting basically weekly treatment since 2016. And so I just wanted to highlight her story there. And then really just to also acknowledge that I think the dam has been breached. So Chemtrac has, made that little hole in the dam, and hopefully many, many more approvals for uveal melanoma. I should say uveal melanoma. I shouldn't even say metastatic uveal melanoma, because we certainly are also in need of an adjuvant therapy. But I think they're right there, just waiting to also, come through the dam. So I just want to finish up with a big thank you to all of the patients, both here, at Jefferson and elsewhere, to certainly my team here at Jefferson, all of our research coordinators, inpatient, outpatient nurses who, we treated a lot of patients with TEBI here and couldn't have done it without a team effort. And I know that's the truth for other institutions worldwide. So I'm going to keep this questions to ask your provider up, but I think I will get out of.
Yeah, Marlena, can you hear me?
Yes, I can. I'll get out of presentation mode there so I can see the rest of the screen.
Okay, we've got a number of questions on Facebook.
Okay.
Bear with me as people who know me.
Let me know if we want to go through those in order, or if they're highlighted here, I can go through these as well in order.
Okay, I think let's take some of the questions on Facebook. We've got a lot of, so first of all, Marlena, you should go back and read these. There's a lot of gratitude to you in here. I'm gonna get to some of the questions. So here is a question. In the indications for Chemtrac, it says it could be used to treat unresectable primary ocular melanoma. Is it being researched as the possible treatment for primary tumors that are not able to be treated through radiation?
So that's a good question. None of those patients were actually necessarily, I think, included in the original study, but not that we should be doing what cutaneous melanoma does, but if we think about cutaneous melanoma, there was drugs that were also approved for kind of unresectable or metastatic. I think the unique situation in uveal melanoma is most of the time the primary is manageable, whether it be with radiation or the larger tumors, being enucleated. So, I think while that's, in the label as a potential indication, I think we would probably need to have that would probably be very select circumstances. And then also, probably looking at that subset of patients before making that, I think, kind of routine care. I think it would be different if someone showed up with a primary eye tumor and, say, metastatic disease at the same time. But for those of you who are listening who've had uveal melanoma, that when it's in the eye, you can have a lot of symptoms. Sometimes there can be pain. And so, I would worry by just using this drug, especially because we don't see necessarily, at least yet, a lot of reduction in size. I don't know if I would rely on, Chemtrac alone to manage the tumor in the eye.
Great. Thank you. And just kind of going along disease progression, another question on Facebook, and it was actually on your list as well. But what about Chemtrac in the adjuvant setting? So for people who have had their primary eye treated, do not have evidence that the disease has spread, is there indication for Chemtrac in that setting?
So not yet. So a lot of you have heard me as well as others speak about adjuvant therapy and what is adjuvant therapy. And, if we kind of take a page out of the usual playbook, drugs get approved in the metastatic setting first, and then they're moved up. That's how it's gone in cutaneous melanoma, that's how it's gone in breast cancer, lung cancer, a lot of cancers. The problem's been is that we haven't had anything in the metastatic setting to move up and test. So a lot of our adjuvant studies that we've done, have really been looking at drugs, with some other kind of rationale for why we might use them there. So, now that we have a drug that is effective in the metastatic setting, yes, we definitely want to try to move that up and see. Chemtrac, though, obviously is a little bit unique. One, in because of the HLA restriction, and two, in its weekly treatment. So, to give a patient, and the first 3 doses are given in the hospital, and there's a risk for that thing called cytokine release syndrome. So, the thing with patients who are high risk is you're high risk, right? It's not 100% that the cancer is going to come back. So you want to be very careful in designing an adjuvant therapy, an adjuvant trial in that population, because you're potentially going to be giving the drug to people that may not have needed it. So, we've definitely been talking about it for years now, and adjuvant trials in discussion, but so just keep your eye out, because I'm sure it's going to be coming down the pike soon.
Great, thank you for that. And on that note, a question about, is there any movement on the treatment timing? So having spreading out the treatment so that it may not have to be weekly, but having more time in between treatments.
We don't know yet. Again, not wanting to lump uveal melanoma in with cutaneous melanoma. This drug has been tested in cutaneous melanoma, looking at kind of different types of treatment schedules. I think we just don't know yet. I mean, some of it has to do with the mechanism of the drug and kind of its, essentially kind of half-life and how long it kind of keeps the immune system there. So we're not sure. We're not sure if we are definitely going to need it weekly. For now, yes, it'll be given weekly. Patients can skip weeks, so that always comes up. It came up when patients were on the trial. Certainly it's going to come up now that it's more widely used. Well, what if I'm on vacation? What if I'm on vacation for a week? What if I'm on vacation for 2 weeks? What if I'm on vacation for 3 weeks, or something comes up, or I'm ill, or I can't get somewhere, when we were doing this trial, it was like peak Philadelphia events. I think it was like the DNC was here, the Pope was here, there was some other stuff that happened. And there was just some weeks where the patients like couldn't get here. And certainly with COVID, that posed a lot of challenges. And again, the study was done during COVID. So you can skip weeks. We don't like it when patients really skip more than 3 weeks, because there is a concern then the immune system can kind of get recharged again. You may have those more side effects like you had with the first few doses, but there is some flexibility.
All right, that's super helpful. And I think a reminder of just how important it is that all of these treatments are developed with patient input and real lives in mind. And so the more we can get patients involved and giving feedback, et cetera, and the more opportunities for that, it's so important for, this reason and others.
Mm-hmm.
A few more questions here. Let's see. I think you answered this, but maybe I'll pose it to you again because it was on a slide that maybe was a little bit complicated. But the question is, what is the percentage of patients receiving Chemtrac who had at least a partial response to the treatment?
So I can go back and share my screen if that'll be helpful with that chart.
And actually, maybe I can have you answer 2 questions at once, because there's another question asking about what about metastatic disease to lymph nodes? And maybe you could just speak to kind of the different locations and if there was a differential
responses. Okay, so here, this is, again, just the overall responses. So really the overall response was 9% in the Tebi arm. And that was also really the patients who had a partial response. So here you can see the absolute number was 22 and it was 9%. The one patient who here had a complete response only added like 0.4%, which is why you still see it as just a 9% overall response rate. And I don't think we have the data to tell us, in patients who did respond, did they respond in the liver, but not the lymph nodes, but not the liver? Again, looking at response, there's not a lot of response, right? Only 9% of patients actually had shrinkage of tumor. I don't know explicitly those 22 patients if they were more likely to have a reduction in liver or non-liver.
Okay, very helpful. Will you leave your slides up for 1 second? Because there are a few questions about HLA typing.
Okay.
Maybe we can knock some of these out.
Sure. I always find it easier to kind of have that diagram.
Yeah. So question about how do you read the HLA typing? So if you get results and your value says A11, A24, what does it mean?
So,
and,
okay. No, you go.
Can I double it up with one other question? Have studies been started with other HLA types?
Okay, so the first thing I will say is that reading HLA reports is very difficult. There are times where I get HLA reports that I actually have to send to, our experts in our tissue typing lab, because I can't tell if the person's actually positive or negative. I would definitely encourage you to talk to your provider who's ever ordering it. Since we've been participating in these studies, we send in-house test and very specifically ask, tell us in a line, give us the HLA type, but tell us in a line, is this person in caps all positive, or is this person, in all caps negative? So it's very clear. So, if you're in an institution that has the means to do tissue typing, often that's done at places that do things like bone marrow transplant, because like I mentioned, or organ transplant, because HLA is important for those as well. Your provider can actually talk to the lab to get a very clean answer. Sometimes if you go through something like LabCorp, or there's other more private kind of entities that can do HLA testing where you send your labs, sometimes the reports can be more lengthy and a little bit more complicated. And I would just say talk to your provider to help you go through it in case it's not clear.
Great. And then I think you spoke to this a little bit in the presentation, but just thoughts about research into looking at
other, right, other HLA types. So, I think that's, obviously something that I think the company is interested in, certainly, us as providers, and I know the patients are interested in. I'm unaware of any, official clinical trials, that are doing that yet. It would make sense that, they would then expand to the next most common HLA and then the next most common HLA. There's probably about 4 or 5 HLAs, that if you kind of did it to all 5 of those, you could probably capture a good portion of patients. I think the question is, though, does a full clinical trial need to happen again? Like a soup to nuts, take 6 years, to just reestablish it, with a different HLA. So, I know there's been discussion, and similarly to the Adjuvant study, I would just keep an eye out for that coming down the pike.
Great, thank you. I think we'll take a few more questions. There are some really good questions keep popping up. So I want to try and get to all of these. There are a couple of questions about the order of different treatments. So question about, for example, if a patient is not eligible or doesn't have access to this right now and they do other treatments and then they become eligible or they have access, can they come back to this? And then a couple other kind of specific treatments about chemo, embo, first, after. How are you thinking about the order of treatments and how should patients approach this?
So I think you guys have heard me say this before. Anyone who's, participated in any of our other webinars or meetings. And I think this was actually a topic at our last meeting in November, we don't know exactly what the best sequence is going to be. You can certainly get other treatments and then come back to Tebi. Even if you got Ipi-Nivo before, I mentioned and I showed an example of somebody getting Ipi-Nivo after, but you could really have gotten any treatment and then get Tebi. Are there patients where Tebi first line makes more or less sense? Like I showed, we see that it doesn't necessarily shrink tumors. Okay, so if we have a patient with a really large tumor and we're really concerned just about the size, so that happens sometimes in the liver. We do a lot of liver-directed therapy. If we had somebody with really dominant liver tumors that we thought we could maybe better control with chemo embo up front to kind of shrink and control the liver, because we know the liver is like the #1 game, we may offer liver-directed therapy before putting them on Tebi. And someone who comes with very limited disease, who doesn't have bulky tumors that are causing pain or anything like that, we may start with Tebi. And there's not to say that patients in either of those groups, could be swapped. So I think we don't know. It's certainly kind of a patient by patient, discussion. And now that not only do we have Tebi and liver-directed therapies and Ipi-Nivo, I showed you there's I meant to count 20 trials that are potentially, available nationwide. And those are really just US trials that I threw up there. So the name of the game though, and you've heard me say this too, is that, I think it's because, we're not seeing complete responses, right? There was, really one patient that had a true complete response here. That's not to say that there aren't going to be others, that get treated. the name of the game is still lining up therapy after therapy to get us the longest possible survival. It's amazing if you can stay on Tebi for 6 years, like that one patient. But most patients don't stay on for that long. So really it's about lining up therapies. So you always want to be cognizant of starting one therapy that may exclude you maybe from another trial. So we always, when I talk to patients and counsel patients, okay, what to do next, we go through all the options and we say, okay, if we do A and B, will that prevent C and D? Do I do D before B? And then we try to figure it out. So there's probably not one right answer. It's certainly a discussion with your provider.
Great, thank you for that. And I think with the FDA approval of this drug, sometimes clinical trials will have exclusion criteria, which will dictate what treatments you may have been able to have prior to that.
Yeah.
But in the FDA approval, there is no specificity to what treatments you may or may not have had prior to starting this.
No, but there may be the opposite. So a clinical trial, especially a clinical trial now that's written in 2022, or even clinical trials that may get an amendment, may say you need to have failed Tebi. So anytime you have a drug that's FDA approved, where you kind of can expect what to expect, what the results are going to be, it's typically required for you to get that first before going on a clinical trial where you may not know how you're going to respond. So there may be trials, especially for A201 patients, where it says you need to have failed Tebi before you come on this trial.
And that patients would have to sort out with their provider and the person and the team who's running the clinical trial by trial.
Yes. Because again, I think one of the things is with Tebi, logistically, it's also a little bit of a challenge. So there are some patients that may not be able to do it for one reason or another. So there's always going to be little caveats.
And that is actually, I think, sort of the point I want to end on is there's some questions about accessibility. So how to access this medication, where is it available now? Where will it be available? What do patients do? And geographically, are patients going to have to travel for this? Is it going to be only a handful of centers? Can you speak to the accessibility of it?
Yeah, so I think some of that is still unfolding. So speaking for my institution, we've had a lot of experience with this. Once it got FDA approved, we've kind of hit the ground running with kind of getting it through all the committees and everything like that, and should hopefully be good to go in March. I know a lot of other institutions, certainly institutions that participated in the clinical trials, so had experience with the drug, had experience with, watching patients for those 16 hours, managing. I think we'll be the first places that'll be able to offer it. That's not to say that if you're kind of local, provider did not participate in the trial, that they can't also get, Chemtrac in the short term. But, there is a bit of a learning curve. So I do think that it'll probably first be available at, all of the institutions, certainly that participated in the trial, or institutions that participated in the expanded access. And then the hope would be to, eventually have it, everywhere. What we are telling patients now is, even if you say go to an institution, like a more experienced institution, to get those first 3 infusions, because that's certainly, also where a lot of the side effects can happen. And say 2 months from now, it is then available at a more local, oncology office, that transitioning patients from kind of location A to location B, shouldn't be too much of an issue.
And how do patients find those locations A, as you're talking about?
I don't know if there's a curated list anywhere. I don't know if you guys had a curated list. It's, if the clinical trials link is still up on clinicaltrials.gov, it's, all the sites that had the trial. I think our uveal melanoma patients are amazing at social media. I'm sure somebody will curate a list and circulate it.
That's what I was thinking, the clinicaltrials.gov. But I think we as an organization can, at least we can develop a list of the centers where the trial was open and make that available.
Yeah. And patients out there listening, if you're at a site, or you sounds like of a site that's going to be able to offer it, you just put it out there to the universe, and we'll make sure it gets on the list.
Great. And I think, let me just take one last look over at Facebook. I think the last topic that we'll sort of address here, which you spoke to a little bit in your presentation, but just this question about if you're not eligible for Chemtrac, or if you progress. And actually, just one quick question. I think the picture of the patient who's been on the treatment since 2016 really gets to this point. But the main reason that a patient would come off Chemtrac would be progression, right? So a patient can stay on this as long as they continue to respond. Okay. So
Yeah.
once a patient progresses, or for patients who are not eligible, you showed the list of trials, which is just, I mean, phenomenal in and of itself that we have that many trials in this space right now. And I just want to mention that we have the webinar from our patient meeting in the fall that goes over open clinical trials. I think there's probably been 1 or 2 clinical trials added to that list since the fall. And so we are in the process of developing kind of a partner webinar to today's webinar where we can go through the different clinical trials open to uveal patients. So look out for that in the coming weeks. But any other sort of comments, closing comments that you have for those patients who either have progressed on Chemtrac or for whom it is not available?
So, when we were working on Chemtrac, I think we all kind of felt in our gut, like, this might be it. Like, this might be the one that, that breaches the dam, shatters the ceiling, and I think we were all very antsy about getting the approval, because we knew that just getting this drug approved would really open the door to a flood of others. It put uveal melanoma on the map. I think companies that didn't know uveal melanoma was an unmet need, was a disease that needed drugs, were then made aware that, it existed and its uniqueness and how amazing all the patients are. And there's companies now that are calling, that are knocking on the door saying, hey, I have a drug that might look like it'd be, effective in this disease, hence, the number of trials. The other thing is that, and this speaks to the MRF and what you guys did at the, the FDA hearing and everything. I looked it up, and the average time from phase 1 to FDA approval for a drug is something like 10 years. This was like 6, really. And certainly it happens more quickly in orphan diseases and diseases with high unmet need. But, this was paradigm changing. Again, this was a drug with not a super blockbuster response rate, but an amazing survival in an HLA restricted population. And we were able to get approval in a relatively, I know it felt like forever, but in a relatively short period of time. And if we can just kind of use that, and that's why I said, I think in my conclusion, that we've learned a lot from this, we were able to enroll a lot of patients very quickly worldwide, so I think this really, gave us the playbook, for what hopefully we do again and again and again and again, until we have this huge armamentarium of drugs, and regardless of HLA status. So, yeah, I mean, I think I keep using the word amazing. I just don't know any other word. But yes, and we couldn't do it again without, the patient advocacy organizations and all the patients and providers and everybody listening. And yes.
Thank you for that. I would love, on that note, if your slides are handy, if you would pull back up that dam image. I think that
Sure.
is just such a powerful image. And, I love those closing comments. And, just to mention all the patients who participated in this clinical trial, all the patient focus groups who we had, I don't see it quite yet.
Okay, hold on.
All the patients who participated in focus groups, I think, the power of patient involvement, patients participating in clinical trials, everything that Dr. Orloff just mentioned is so, so important, helped us get to this day. It's going to help break down the entire dam so that it's not a rush of water, but it's water flowing freely. And just thinking back to Dr. Flaherty's comments about where we are in skin melanoma and where we will be hopefully very shortly in uveal melanoma. Dr. Orloff, as you pointed out, the speed at which this can happen is really in our hands as patient advocates and dedicated clinical advocates and researchers. And we know in the uveal melanoma space, we have Dr. Orloff and so many others who are so dedicated to this patient community, and we can really make a difference, and we can push this to happen. So we are so excited for this step. And thank you so much, Dr. Orloff, for helping us learn so much about Chemtrac today. This is just such an historic moment, and we know our work is not done, and we will push through brick by brick to break down that dam. So thank you so much for joining us today. And please feel free to email us if you have additional questions that you think about later or that we didn't get to today. Feel free to email us curom@melanoma.org, and we'll do our best. If we get a number of questions, we can try and come up with a Q&A. We can try and develop a list of centers who participated in the clinical trial, if that would be helpful to patients. And then stay tuned for a follow-up webinar in the coming weeks where we dive a little deeper into the other clinical trials that Dr. Orloff presented today. So thank you all so much, and we look forward to being in touch.